Good afternoon. Good afternoon, everyone. First, let me say thank you for attending the 7th Annual Cure Corridor Luncheon, Advancing the Cure. I am Marion Kelly. I'm Director for the Office of Community and Business Relations at Mayo Clinic, and it's my distinct honor to serve as today's Master of Ceremonies. As many of you know, Mayo Clinic is a nonprofit organization committed to clinical practice, education, and research. We're committed to clinical practice, education, and research, and inspired by hope and change in our industry. That's why we are supportive of the Cure Corridor. As you'll learn today, the healthcare and bioscience industry is changing, and it's changing fast. The accomplishments happening right here in Scottsdale are shaping the way people around the world are giving and receiving treatment. Today's event is an opportunity to celebrate all that our community here in Scottsdale is doing to advance the cure. Now, without further ado, let's get the event kicked off. Ladies and gentlemen, please welcome Mayor Jim Lane.
Well, thank you, Marion, and thank you all for being here. It's wonderful to have you here. So welcome to the 7th Annual Cure Corridor, and it's great to be back with you to celebrate another year of amazing growth and innovation taking place right here in this industry, right here in Scottsdale. We have an impressive lineup of speakers, but first I want to recognize my council colleagues joining us here today. We have Council Members Suzanne Clapp, Virginia Cordy, Guy Phillips, and Vice Mayor Kathy Littlefield. So welcome them all. We're particularly glad, too, to have Senator Kate Brophy-McGee here to join us with us as well. First, I'm pleased to thank Cigna, a loyal title sponsor, for their instrumental support in making sure the Cure Corridor mission to build a stronger and more collaborative bioscience and healthcare community is a success. We do appreciate it very much, and so thank you, Cigna. Our partners in the Cure are Honor Health and the Mayo Clinic. Both are absolute giants in our community, and I thank them for their generous ongoing support of the Cure Corridor. So thank you to both of them. We have a wonderful group of table sponsors who also help make today possible. Arizona Bioindustry Association, Arizona Public Service, Arizona State University, the Honor Health Foundation, Arizona Bank and Trust, Banner MD Anderson, Primus Pharmaceuticals, Systems Oncology, Greyhawk Development, ASU Mayo MedTech Accelerator, and Closys. Thank you all for your generous support of the Cure Corridor and your continued commitment to our community. Please. And a special thank you to Mark Stanton and the Scottsdale Chamber of Commerce, who do so much to help us with putting on today's event. Please, let's give it a hand for the Commerce as well. We have a terrific program lined up here for you today, and it's my pleasure to have Dr. Paul Laikind here to discuss his journey to find the cure for type 1 diabetes, as well as some wonderful rapid-fire presentations. You know, when we started the Cure Corridor effort with the first event over 7 years ago, my goal was to shine a spotlight on all the accomplishments happening right here in Scottsdale in the life sciences and healthcare industries to foster some new tech industry relationships. But the growth and the connection have exceeded my expectations. Practically weekly, I learn about some new development, investment, or collaboration attributing to our local bioscience business or related tech community. From Honor Health's groundbreaking, literally and figuratively, the Neuroscience Institute, a collective connective of neuroscience services that is under construction right now, to ASU and the Mayo's new medical business school, to the startup culture and VC investment we are seeing at SkySong and across the city, this industry has flourished. It is seen by some seeking to be here in Scottsdale as a budding global bioscience hub. We're trying to condense all of this news and its economic impact into a short video, so please turn your attention to the screens right now for a short video on the Cure Corridor. You know, as I've said before, and actually shown there on the screen as well, the Cure Corridor is much more than a geographical location. It's a culture and an environment. And I thank each and every one of you for being here with us today as being part of it. We have a packed agenda this afternoon, so we'd now like to take a very short break and be back momentarily with the rest of the program. Thank you.
To kick things off, let me introduce you to our first presenter, AdviNow. Please help me welcome to the stage for his 5-minute rapid-fire presentation, Robert Backey, Director of Operations at AdviNow. Robert.
Again, I'm Robert Backey with AdviNow. I'd like to thank Mayor Lane and Scottsdale for inviting us to be part of this. Cure Corridor's got some incredible stuff going on. If you look at the companies that are being built, the healthcare stuff that's coming out, it's absolutely amazing what is going on in this area that we're all located. Companies like ours are all focused on this old triple aim of improving the experience, the healthcare for the patient, and lowering the cost. As a company, we believe that taking modern technology like artificial intelligence and augmented reality is going to help us accomplish that. So that would help. So what we've done is our founder came out of the self-driving industry. James Bates was part of the stuff that was helping Waymo and Tesla and other companies build their self-driving cars. We've taken that technology, applied it to the patient journey, whether it's on the technology side at home, as I'll show you, or in the clinic, and are able to help improve the whole process for the patient. But one of the things that we did find through this whole process is that burnout with doctors. Doctors, as Forbes reported, the biggest issues they're facing now is they're spending so much time in front of a computer. They're not spending time seeing the patients. In fact, New York Times just reported that for primary care, over half of a doctor's day is spent charting in a medical record system. Again, not seeing patients where they need to go. We take our artificial intelligence and augmented reality platform, walk the patient through, and by the time the doctor actually sees the patient, we've done 80 to 90% of the charting for the doctor based on what the patient has told our system. What does that mean for a doctor? They're not spending half the day seeing patients. They're spending 3/4 of the day, or doing charting, they're spending 3/4 of the day to 90% of the day actually seeing patients, diagnosing. I mean, that's where you want your doctors to be, not on a chart. So how do we do this? We start with the patient either at home or in a clinic. We give them a tablet, or they do it on their mobile device or on their computer. They register for their appointment, all their demographics. We take the social history. Those forms you all fill out whenever you go in, they give you the clipboard and you just check no, 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 no. We do that on the tablet. No more forms in the clinic. We then ask, why do you need to see a doctor? Get their chief complaint, they put it in, and now we start asking questions using our artificial intelligence to intelligently ask questions that pertain to their chief symptom. 15, 20 questions, 3 or 4 minutes later, we've got a pretty good idea what's going on with the patient. In integrated healthcare systems that have multiple ways to see patients, we can route the patient based on that feedback, whether it's to a telemedicine visit that would actually work for them, or to a retail clinic. If you've been to the Safeway out in Frank Lloyd Wright or Pinnacle Peak, there's a retail clinic that uses our technology in there. Or to an urgent care, or to the primary care, or for those who need it, straight to the emergency room. It's all about properly routing a patient. They show up at a clinic, whether it's retail or urgent care. They walk in, tell the front desk why they're there. They sit down at one of our augmented reality-infused kiosks. Medical assistant or the patient can do it himself. We take the basic vitals. Plus, when appropriate, we take imaging of the inner ear, the nose, the throat, heart and lung sounds, so that a provider, whether they're located on-site or remote, can then go into our portal, review all the documentation from the patient, all of their questions that we asked them, all the answers they gave. They can look at the images. So a strep throat patient, you have the images. We've already asked to have a strep throat test done. Results are put in the system. By the time the provider sits and looks at that screen, we pretty much know what's wrong with the patient. They look at it, review the results, and then see the patient, whether it's in telemedicine or an in-practice visit, and sit there with the patient. Rather than asking questions, they're able to diagnose, treat the patient right then and there in a few minutes. What do we find from patients? They actually find that this workflow and the results from this workflow are some of the best exams they ever had. Why? Because the provider is not spending time asking them questions they've already answered. It's sitting there diagnosing. You've got strep throat. We're going to give you amoxicillin. We want you to go rest, get some sleep. You come back in 3 days. You have any questions? Thank you. Done. Off we go. For providers, for a clinic, double to triple the throughput for a doctor. Not burning the doctor out, because burnout comes from charting, allowing them to see more patients. So where does this all go? We look at the future of what's gone with Waymo and Tesla, with all of us able to drive our cars by sitting behind the wheel and actually not doing anything. We lead the way in using artificial intelligence to help diagnose the simple things. We're not looking to replace a clinician. We can never do it. We want to augment the clinician. Take things like strep throat, take things like a UTI that we don't need to have a provider do hands-on, diagnose, provide an automated treatment method and path for the patient so they don't even need to go see a doctor. We can treat them effectively from home. What does that do? Patient care gets better. We reduce cost, and it's a better overall patient experience. Again, we're AdviNow Medical. We're based in Scottsdale in the Cure Corridor. We are the future of healthcare, and we're doing it today. Thank you. Thank you.
This is information that you can take back and make sure that you're networking here, because the relationships that you build here could last a lifetime. You're hearing new information, new technology, and we hope that you can apply that technology. I'd like to introduce to you and present to some others that may have met Dr. Amy Sarin. She is an internationally renowned neuropsychologist, stress expert, award-winning tech inventor, and not to mention best-selling author. She conducts neuropsychological assessments and provides treatment for children and adults with issues including ADHD, anxiety, depression, traumatic brain injuries, and life challenges. She's going to tell us more about her journey with Touchpoint Solutions and a product that I feel all of us can benefit from. Please welcome to the stage neuropsychologist and inventor of Touchpoints, Dr. Amy Sarin.
Thank you so much for having me. This is really exciting time in the world because our neuroscience is advancing very, very rapidly. And what that means is that all of you have a few things that you could be doing that can change your lives profoundly while you multitask. And I'm here to tell you a little bit about that today. The company that we're focused on today came out of my clinics called the Sarin Center Clinics. We have three, one in North Peoria, one in North Scottsdale, and one in Gilbert. And we see it's about 6,000 patients a year. We do cutting-edge treatments like neuropsychology and neuromodulation. And it's interesting because in having something called electroencephalograms now on an outpatient basis, we can actually track the human stress response in real time. And we can see the effect of immediate technology and treatments in people's brains. And it's very, very great because what we can do is really quantify what's going on. And back in about 2014, I was tasked with making sure that certain individuals in the foreign military elite never got PTSD. I thought that was impossible, but then I was told it was my job, and so I set to figuring out how we could actually prevent PTSD and doing it in ways that are non-invasive, cost-effective. And then that group later went into responding to the Ebola crisis, and my job was to help stabilize villages post-Ebola and make sure that they didn't get PTSD and they could restabilize as quickly as possible. What I found was, in my neuroscience journey, that there is a network in your brain that actually controls stress, and it's a preconscious network. That's good news for you, because that means you don't need to think your stress down, and this is why you can't do that. So, fast forward to today, we have a very, very technologically advanced waveform in two small wearable devices that have been on the market since 2017 called Touchpoints. And for the cost of less than one therapy appointment, you now can have 62% less stress anytime you want, and the effect happens in about 30 seconds. I'm just going to pause so you can just take that in. Just for a moment, okay? And I know that sounds too good to be true. I have them if you want to try them afterwards. And the good news for me is that it works immediately so people can tell the difference. We also have research backing this up. Since 2017, we also have won several international awards, including 2019 Forbes Best Technology and a 2018 Edison Gold Award. So this is where acupuncture and yoga and exercise were back in the 1980s. But the advantage is that I am not going to tell you that to manage your stress, you have to leave what you're doing and add 2 hours to your day. Okay? So in order to explain this, I came out with a book that actually recently hit #1 on Amazon, bestseller for preventative medicine. And in the book, it explains the new neuroscience of stress and what we understand to be true. What we used to think is that if a snake comes in, we recognize the snake, think about what to do, and go into fight or flight. Wrong. We actually are in fight or flight before you consciously recognize it because consciousness is too slow of a road for survival. Now, we can hack into that system through senses, and we can do it through sound, we can do it through touch, but the waveform in the Touchpoints product is actually the fastest way that I could figure out to do it, and it actually reduces stress 62% in 30 seconds, and that's double-blind controlled research. Now, I want you to take one step back and think about your stress switch and how it operates. Right now, you're on a scale of 1 to 10 somewhere, right? Now, maybe you're pretty low stress, or maybe you just had an automatic thought that you forgot to do something, or how many emails are going into be in your inbox when you get back to work, or something like that. Your stress switch is fluctuating all the time, whether or not you're aware of it. And keeping a low stress switch is actually the key to whether or not you're focused or distracted, patient or irritable, kind or selfish. And so it behooves all of you to pay attention to your stress switches, because when you moderate that, when you're in a regulated state, you're better for yourself, you're better for everybody else, and you don't have to lay in bed at night with that monkey mind going, not being able to sleep. And I know you all know what I'm talking about, right? So, my last slide is the research, so that you don't think this is snake oil. This is one study done by Uninove in Brazil at the University of Sao Paulo. And what they found was that after the Trier social stress test, where they stress people out and have them actually do mental math in front of people, can you imagine? Mental arithmetic in front of a bunch of stoic judges. The blue line is the cortisol levels afterwards. If you see those 4 stars, for those of you who are researchers, you know that that is a p-value of 0.00001, which means about a less than 100,000 chance that it's actually the placebo effect, okay, with just a sample of about 80 people. And in the placebo condition, the cortisol stayed high. So now we can show that you can go through a very incredibly stressful situation and at the end of the day be less stressed than when you showed up that day. I think that's incredible, and I think it can change lives. And this is part of the reason why Touchpoints have sold over $5 million in the consumer market in 2017, and the applications are endless. We have anesthesiologists using them to stabilize blood pressure before people go into surgery. We have autistic children wearing them in school, and it's reducing their self-harming and self-disruptive behaviors by 72%. We have people falling asleep at night without Ambien. We have people ditching their Xanax because now they have something that lowers their stress in real time. And we can add this to sort of the complete breakfast of treatment of things like PTSD, anxiety disorders, and for the general 83 problems that we all suffer with. So I want to thank you so much for your attention. Some of you here do have the book. I hope you enjoy that. And I hope that you're excited about the new advances in neuroscience, and I hope you take it upon yourself to make your lives as great as they can be. Thank you.
I'm pleased to introduce today's keynote speaker. Paul Laikind has over 28 years of leadership experience in the biotechnology and life sciences industry. He currently serves as president, CEO, and a member of the board at ViaCyte, a leading company in the emerging field of regenerative medicine. Prior to joining ViaCyte, Dr. Laikind served as the Chief Business Officer and Senior Vice President of Business Development at the Sanford Burnham Medical Research Institute, where he established a number of licensing and strategic partnerships with large pharmaceutical organizations, including collaborations with Pfizer, Centers for Therapeutic Innovation, and Ortho McNeil Janssen Pharmaceuticals. Please help me to welcome to the stage President and CEO of ViaCyte, Dr. Paul Laikind.
Well, thank you, Mayor Lane. Thank you, the City of Scottsdale, for inviting me out here today. I actually have 2 brothers who live here, so I feel fairly connected to your city, and really excited to hear everything that I've had breakfast with the mayor this morning and learned a lot about what's going on in the city, and it is really exciting. So what I'm here to do is tell you about some of the work we're doing at ViaCyte, a really exciting company that I joined recently, or actually not so recently. But we're working on cell therapy for major diseases, and our major focus right now is actually to deliver a cure for type 1 diabetes. So you heard a little bit about my background, and I just wanted to fill it in a little bit more. So I came to San Diego back in 1979 to pursue a PhD at the University of California, San Diego. And at the time, the biotech industry was very young. There were only a few companies out there. And so I've had the pleasure of being involved in the evolution of that industry in San Diego to where we have over 600 companies in the field. In 1986, after getting my PhD, I left the university to start one of the early biotech companies in San Diego. I went on to be the founder of 2 other companies in San Diego. All 3 of these companies went public and were eventually acquired. And then, as mentioned, I did a brief stint at Sanford Burnham Medical Research Institute, really to help them to get more translational. And then about 7 years ago, I joined Biosite. And this is actually the first company that I've run that I didn't actually start. And I have to say it's probably the most exciting company that I've worked on. And it goes well with the theme here of cures, because why it is so exciting is it really gave me an opportunity to work on a company that wasn't just working to impact the disease, but actually to cure it. I've also been very involved in the innovation community in San Diego. I've been on the board or chair of both Connect and Biocom, 2 organizations that help innovation there, and also I'm on the board of the Alliance for Regenerative Medicine. So Biosite's vision is pretty straightforward. We're working on cell therapy-based cures. This is, as it was mentioned, a regenerative medicine approach, and really we've been pioneering this field for well over a decade. The initial focus is on a potential cure for type 1 diabetes, as I mentioned. However, the platform itself has applications in many other technologies, and we're really excited about where we can take this. But today I'm going to talk more about the diabetes approach. So many of you, probably everybody in here, knows somebody with type 1 diabetes, and there's type 1, type 2, very different diseases. But type 1 disease is an autoimmune disease where that autoimmune reaction leads to destruction of a cell type called the beta cells in the pancreas. These beta cells are part of a structure called the islet. There's other cells that are important, but the beta cells are responsible for regulating the release of the hormone insulin. Insulin regulates blood sugar levels, and it's essential for life. In fact, 100 years ago, type 1 diabetes was a fatal disease. It was pretty indiscriminate, and back then, a patient with type 1 diabetes, their life was heartbreakingly short. This is a child back 100 years ago, just before the introduction of insulin, and you can see kind of wasting away. And children, typically when they were diagnosed with type 1 diabetes, would die within the first year after diagnosis. Adults might go a few years beyond that. This all changed in 1921 when 2 Canadian physician scientists, Dr. Frederick Banting and his then student assistant, Charles Best, successfully extracted insulin from dog pancreases and showed that it was effective for restoring the regulation of blood sugar. This important finding earned Dr. Banting a Nobel Prize, and it quickly led to the use of insulin in humans, changing type 1 disease from a fatal disease to a chronic disease. So that was a major advance. And you can see that in this next slide. This is that same child now that you see that was wasting away just a few months after going on insulin therapy, and you can see a remarkable change. So clearly the isolation use of insulin was momentous and historic. Still, 100 years later, we're basically doing exactly what we did 100 years ago. We measure blood sugar levels and we administer insulin. And while that restores life and changes it from a fatal disease to a chronic disease, it still is a disease that has many issues. One, it has a big impact on quality of life. So in order to manage your type 1 diabetes, if you have this disease, you have to carefully control and monitor what you're eating. You have to watch your exercise and keep track of it. You then have to measure your blood sugar levels on a regular basis, either with a finger stick or with continuous glucose monitors. And then you have to administer just the right amount of insulin, either through an injection or more recently infusion pumps, to get it just right. And you often don't. There's a lot of hypoglycemia. A lot of hyperglycemia goes along with it. On the next slide, I'm going to show a quick video, and it's just a caregiver talking about what the disease means to him that I think is really interesting.
It's a fatal disease. I call it spending your life not dying.
So, spending your life not dying, that's really an amazing statement about this disease, and it really is true. Every day you have to think about this disease, and you have to treat it appropriately. And if that's not enough, the immediate day-to-day impact of this certainly has an impact on quality of life, but it also has a longer-term impact. Patients with type 1 diabetes have a propensity for long-term complications, so it's kind of adding injury to insult. So patients with type 1 diabetes have an increased risk of heart disease, stroke, blindness, kidney disease, ulcers, and even amputations due to vascular disease. But there's hope, and that's where we come in. About 20 years ago, a group of scientists led by Dr. James Shapiro up in Canada developed a technique using islet tissue that has been isolated from pancreases taken from cadavers. And they showed that if you isolated these cells and you gave enough of them, they infused them in the liver portal vein of the patients, you could essentially functionally cure the disease. There are patients that were treated almost 20 years ago that are still completely insulin independent. And typically patients that receive a cadaver islet transplant can go about 5 years or longer without any need for insulin. So it shows that if you could replace those cells that are lost to the autoimmune disease, you can potentially cure this really detrimental disease. However, there's a very limited supply of pancreases that are suitable for doing this. There's only about 1,500, believe it or not, available in the US each year. Most of those go to dual kidney pancreas transplants. It's pretty costly. It requires lifetime use of immunosuppression. And as a result, there's only been about 2,000 patients treated over the last 20 years with cadaver islet transplant. And that's in a population of millions of patients with type 1 diabetes. So that's where ViaCyte comes in. So ViaCyte is leading the way to a functional cure for type 1 diabetes using a stem cell-derived islet replacement therapy. I always quickly point out that we don't treat patients with stem cells. You hear a lot about that. We start with a stem cell, and then we drive that up to a pancreatic cell, and we use that to treat the patient. But we're building on this proven ability to cure type 1 diabetes with a cell replacement therapy. We have multiple generation of products, which I'll introduce you to in just a minute, that are addressing increasingly larger sectors of the diabetes market, not just type 1, but even potentially insulin-requiring type 2s, which is a much broader population. We have already shown in the clinic, we're in clinical trials with 2 of our programs right now, and we've shown in the clinic that the cells, the replacement cells that we produce, are effective at replacing those beta cells and producing insulin. And we're working with world-class partners. We work with W.L. Gore & Associates, which has a big presence in Flagstaff, and also with CRISPR Therapeutics, one of the top gene editing companies. I'll talk more about that in a minute. We've also had tremendous support, about $80 million of support from patient advocacy groups, including JDRF and the California Institute of Regenerative Medicine. And then we have some great investors, including Bain Capital, TPG, RA Capital, and Sanderling. So we have been leading the way in this. We were the first company or first group ever to publish on the production of beta cells from pluripotent stem cells. We were the first group to show that those cells could then be put into an animal model and be curative for an animal model of type 1 diabetes. And we were the first company to move a product based on an islet cell replacement technology into patients. We have an extensive IP portfolio. We actually acquired, the only other company that was really competing with us for a while was J&J with a division called Betalogix. We acquired the Betalogix division about 4 years ago and moved all their IP into ours. They own about 20% of us now. And our IP portfolio is almost 600 issued patents, so we pretty much dominate the space. So what I'm going to show you now is a brief animation that will give you a better understanding of the products than I can probably do, and then I'll come back and talk about them in a little bit more detail.
What if, instead of injecting insulin all the time, Type 1 diabetes can be treated by simply replacing the missing beta cells. This is the idea behind ViaCyte's groundbreaking technology. ViaCyte has a family of product candidates in development, each of which consists of an implantable pouch containing cells designed to behave like those in the pancreas. Based on ViaCyte's leading research, the cells are carefully and fully differentiated from stem cells into precursor cells of the pancreas. Since these cells are made from a stem cell line, there's a virtually unlimited supply available for manufacturing ViaCyte's product candidates. Once differentiated into pancreatic precursor cells, the cells are loaded into a pouch, which is then implanted under the skin. After implantation, these cells are designed to mature into all the types of cells in the endocrine pancreas. Once matured, the implanted cells have the potential to monitor blood sugar and respond with secretion of insulin and other hormones to control blood sugar levels. ViaCyte's product candidates differ based on the type of pouch or cells. For Pack Direct, the pouch is designed to allow blood vessels to enter the device and directly interact with the implanted cells. Because the implanted cells are not hidden from the immune system, patients who receive Pack Direct must take immunosuppressants, making it suitable only for people with high-risk type 1 diabetes. For all insulin-requiring diabetes, ViaCyte is developing PEC NCAP. For PEC NCAP, the pouch is designed to fully contain the implanted cells, but still allow vital nutrients and proteins, such as oxygen, glucose, insulin, and other hormones, to travel between the cells inside the device and the blood vessels, which grow along the outside of the device. This device is also designed to prevent immune cells from directly contacting the implanted cells, so they may thrive and function without provoking an immune response or being destroyed. For PEC QT, cells will be specially engineered to avoid provoking an immune response, potentially eliminating the need for immunosuppressants. The cells will likely be housed in the same type of pouch used for PEC Direct, which allows direct interaction between blood vessels and the implanted cells. But because the cells are designed to be immune evasive, they are not expected to be rejected by the immune system. With almost 2 decades of research and leadership in the field, 2 product candidates in clinical trials, and a third in preclinical development, ViaCyte believes it has the key elements needed to produce transformative therapies for all people who require insulin to treat diabetes.
Okay, so that gives you a good understanding of the products. Let me give a little bit more detail on how this works. So we started with a pluripotent stem cell bank. It's actually an embryonic stem cell line that we isolated almost a decade ago, and we have thoroughly tested it. It's been reviewed by regulators at the FDA, Health Canada, European regulatory authorities, and all of them have given it a green light as a starting material to manufacture our cells. It does represent essentially an unlimited supply of cell material. We have master and working cell banks. So when we're ready to manufacture, we take a vial of cells out of the working cell bank, and we go through a scaling process going from millions of cells to billions of cells in about 2 weeks. And then we go through a 12-day differentiation process to go from that stem cell up to a pancreatic progenitor cells that's destined to go on to form all the cells of a normal human islet. That 12 days took about 7 years to develop, okay, and represents hundreds of patents on this technology. Once we're ready to treat, we can then cryogenically freeze those or stable. We can then do all the release testing. And when we're ready to treat a patient, we take a vial of cells out of the freezer, we thaw it and culture it for 3 days, and then we load it into a device. It's a very small, thin device. Actually, I have one in my pocket. And we load it into this device. You can think of it like a tea bag. And then it goes out to the clinic, and it's implanted under the skin subcutaneously. The cells are designed to stay in that device, and then they get vascularized and differentiate over a several-month period to become all the cells of a normal human islet, producing insulin and glucagon and all the factors. So that's the goal. Of course, saying all that is easier than making it all happen, but we've been working on this now for quite some time. We have, as you just heard in the video, we have 3 products that we're working on: PEC Direct, PEC and Cap, and PEC QT. You can think of these as first generation, second generation, third generation products. Although PEC and Cap was the first in the clinic, and I'll explain that in a minute. But the one thing that is key here is all of these deliver the same cells, okay? These PEC-01 cells, these pancreatic progenitor cells. They deliver them somewhat differently. In the case of PEC Direct, it uses an open device. We put ports in it. It allows direct vascularization. That has certain advantages. It overcomes any foreign body response that could interfere with the implantation. It gives better dosing and longevity potentially by having that open device. But the downside to it is because it's open to the host cell immune system, you have to use long-term immunosuppression. So it's only targeting the high-risk type 1 diabetics. The PEC and Cap uses a fully encapsulated system, no ports, and instead uses diffusion across that membrane. It requires a vascularization on the surface of the membrane rather than in the lumen of the device. And as a result, it avoids the need for the long-term immunosuppression. So it opens it up to all type 1 patients. And then a really exciting approach that we're working on now in collaboration with CRISPR Therapeutics is to take our stem cell line and use gene editing technology to knock in certain genes, knock out certain genes in such a way to make that immune evasive, and then be able to use that in that open device and get the advantages of that. So, as I said, the common thread on all 3 of these products is the cells, and we have good data now from patients in the clinic. I'll just show you some data on this, that when these cells get effectively engrafted, when they get well vascularized under the skin in the patients, they do what they're designed to do. So what's shown here over here, we're seeing histology. We put in small devices and take these out and look to see how the cells are doing. And here you can see the cells are thriving, and this is staining them for insulin production. And this is typical of what you'd see in a normal human islet in terms of the number of beta cells. Very importantly, we can also show now that when we get the better engraftment, patients are now who came into the study completely unable to produce insulin are now producing insulin 9 or 12 or 15 months afterwards. So we're really excited. We're on the right track. We still have more work to do. In terms of each of these programs, as I said, this is a little bit of a history that gives you a feel for what it means to pioneer a field. So talk a little bit more in detail about PEC and Cap, which is our second generation product for all patients. As I mentioned, this was the first one to go into clinic. And the reason was if this worked right out of the box, then you wouldn't need the one for the high-risk patients. You could use this for all patients. But when we were getting ready to go into the clinic with the cells, we had data from 2 different animal models. One was a mouse model where you had what's called a mild foreign body response, and it worked beautifully in that. And many other groups had done the studies and shown thousands of animals had been studied, and it worked perfectly. However, we also had data in a rat model that had a very aggressive foreign body response, and it didn't work well in that at all. And what happened is you got what's called foreign body giant cells, which is what those arrows are pointing to, that basically lay down on the membrane and interfere with the ability to get that vascular network. So when we were getting ready to go into clinic, the question was, were we more like the mouse or more like the rat? Who wants to guess? Most people say, obviously, we're a bunch of rats, and they're right. So basically, we went into clinic with our product, and we asked the question, are we seeing it more like the mouse or more like the rat? We showed that it was safe and well tolerated. We showed that the device protected the cells from the immune system and all, and we showed that we could get regions where you get good vascularization and the cells would thrive in those regions. However, in most cases, what we saw was what we saw in the rat, a very aggressive foreign body response that limited the ability to get that vascularization. So we did 2 things at that point. One, we moved forward with PEC Direct because it allows direct vascularization, and it just basically goes around that foreign body response. So we moved that into clinical development. And then second, we formed a collaboration with W.L. Gore and Associates. The membrane material we use in our device is expanded PTFE. It's a medical grade plastic known by the trade name as Gore-Tex. And most of you associate Gore-Tex with outerwear and clothes and such, but about 50% of Gore's multi-billion-dollar worldwide revenue stream comes from implantable medical devices using this material. And so we formed a collaboration with Gore to work with us to evaluate this membrane material and modify it in such a way that would resist that foreign body response. I'm pleased to say, after about 3 years of working together and screening dozens of different membrane configurations, we now have identified several membranes that do appear it now work really well, as you can see in this slide. Here's that same rat model. Now we're seeing good cell survival out at 38 weeks. We're seeing good C-peptide, which is a biomarker for insulin production, so human C-peptide production. It's 5 to 6-fold improved over the original membrane. So we are now back in the clinic with PEC and CAP. Right now we're doing studies with the sentinels to show that we are translating what we just saw in the animal models to the humans. We're very excited about this, and we're looking forward to moving PEC and CAP back into therapeutic studies next year. So the summary on the PEC and CAP program is that the early clinical results established some important proof of concepts. We identified and characterized that we did have a very aggressive foreign body response, and we used the clinical insights in collaboration with Gore to develop device membranes that now are resisting that foreign body response, and we're back in the clinic. In the meantime, though, while we were working on that, we moved forward with PEC Direct. And that's because we knew from our studies that it was not impacted by that foreign body response. It worked just as well in the rat model as it did in the mouse model. So what we're targeting with PEC Direct are the 10% of type 1 diabetic patients who are at high risk for acute complications. These are typically patients with hypoglycemic unawareness or brittle diabetics, and they end up on the emergency room on a regular basis. They're managed by a small number of specialists. It will require immune suppression, which is why we targeted the high-risk group, but it represents a very significant commercial opportunity and addresses a very important need. So most of our data on the production of insulin comes from PEC Direct, and this slide is just to say that this is some data from one of our better patients where we're getting up to therapeutic levels of insulin and showing that it's glucose responsive. So it's, again, further confirmation that those cells do what they're designed to do when they're appropriately implanted in patients. So our clinical progress on PEC Direct, we've shown that the direct vascularization is achieved. We've showed that the immune suppression protocols we're using do protect the grafts. We've shown that the cells, when they effectively engraft, do what they're designed to do. We are planning to get what's called a Regenerative Medicine Advanced Therapy designation from the FDA and are gathering data for that now. That could help accelerate the development of this program. We are still optimizing. One of the unique features of human cell therapy is you can't really optimize in animals. These are human cells that we put in humans. When we take those cells and we put them in an animal, that's crossing a species, and you get a very different response to that. And so most of our optimization takes place in the patients, and the FDA has had to work with us, or we've had to work with the FDA to allow us to do this type of optimization in the clinical trials. But we're making good progress, and we're looking forward to potentially demonstrating the initial efficacy over the next 12 months or so. So this is just summarized in this slide that shows the various steps along the way we're going to get to that pivotal phase 3 trial, which we expect to happen as early as 2021. So where we are with this program, we have clear clinical endpoints. We expect the RMAT designation. We have the preliminary clinical evidence showing that we get the active substance, the PEC-01 cells, effective in these patients. We're focusing on improving the effective engraftment, and additional clinical optimization studies are ongoing. So finally, I'll just leave you with our latest program, what we call PEC-QT. And this is creating an immune-evasive version. So this summarizes what we're doing in this area. We're taking our stem cell line, this pluripotent stem cell line that we've bank that we've thoroughly tested. It's probably one of the most tested stem cell lines in existence at this point. It's been approved by the regulators. We take that and we knock in certain genes and we knock out certain genes in order to make it immune evasive. And then the goal is to show that they remain pluripotent. And if they do, then you can make our pancreatic cells from that and potentially then use that as a treatment without the need to go to extra measures to protect those cells from the patient's immune system. What's really exciting about this is this not only opens up for diabetes, but if you have an immune-evasive stem cell line, theoretically you can make any cell in the body with that. So it opens up many other applications for this technology, and we have patents on many of these. So we're already having great success in this program. We're doing it with CRISPR Therapeutics, which is one of the top gene editing companies in the world right now. And we just presented results on our first edited cell line at the European Association for the Study of Diabetes meeting. And we showed that we were able to knock in certain genes, knock out certain genes, and then showed that it is immune evasive in an in vitro assay, and we've shown that they remain pluripotent and we can make the PECO1 cells from it. So we're off to a great start. We just started this program with them about a year ago, but already reporting out the first success from it. We have other constructs that we're studying, and we hope to be moving this into the clinic before too long. We're already discussing IND-enabling studies. So as I said, we have an expansive IP portfolio that covers all the various aspects, going from a pluripotent stem cell up to these pancreatic cells. It covers manufacturing, covers device and delivery technologies, and it includes almost 600 issued patents and over 100 issued patents in the U.S. alone, and we still have about 400 pending applications. So to summarize, we have 3 programs. We've shown that the PECO1 cells are effective, so you can check that one off. And now it's a matter of delivering them in an effective form for each of these programs. From a financing point of view, we've been venture capital financed. Our latest round was a Series D financing of about $80 million at the end of last year with Bain Capital, RA, TPG, and Sanderling. We have collaborations with CRISPR Therapeutics and with W.L. Gore. We had some funding from them last year too, so a total of about $105 million. And over the years, we've had almost $90 million in patient advocacy support. So we think we have the products, we have the plan, and I know we have the team that can deliver this cure. We still have a lot more work to do, but we're really excited about it, and we're really thrilled to be on the cusp of potentially changing the way this disease has been managed for 100 years. So what we want to do is we want to go from the way it has been managed with injecting insulin and trying to get it just right to a point where we implant a patient with these devices and they can forget about their disease for years. Thank you.
I'd also like to ask Marion Kelly, Robert Becky, and Dr. Amy Sarin to come to the stage as well, just for a small presentation. Well, I hope everybody certainly enjoyed and learned something here with us today. I mean, a day without learning is a lost day in life's progress. So I hope we all had a good time doing it, voluntary learning. But I do very much appreciate you all joining us in this, I think, a great program we've had here for a number of years now in advancing this industry here in Scottsdale. So thank you very much for being here, and continue to have a good day today. Thank you.